No, ma'am. Employees didn't join.
Okay. We wait for another 2 minutes and then we initiate.
Okay, ma'am.
About what we are going to discuss today, what this session is all about. Yeah, can you tell me what today's session is all about?
Environmental management system.
So what's your thought on that? What's your opinion on that?
We should not pollute the environment.
Okay, so how will you ensure that you will not pollute? Very interesting intent. We all have the intent that we should not pollute the environment. What are you going to do about it? Do all of you agree with her? I mean, she's been good enough to open the floor for discussion. Do all of you agree with that or you don't?
Yes. Yes, ma'am.
Yes or no? I want an answer.
Yes, ma'am.
Otherwise, I'll ask
Yes, ma'am.
one by
Yes,
one.
ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
You all agree with me. So how do you think as an industry, you have been studying now from past couple of weeks, how do you think as an industry we should handle this collectively? Or how has the industry handled this collectively? How has the industry handled it collectively? Not only that, even in our house also, how have he handled it collectively? How has the world handled it collectively? You are aware of it, but because you're so focused on the chapters that is being taught that you have to bring it back from the back of your mind to the front of your mind along with quality.
Decontamination, ma'am.
Safety, sorry, please.
And we should do a decontamination process to eradicate the problems will come in the future, ma'am.
Decontamination of what?
The waste disposals.
Okay. So as I said, in the industry, quality, safety, environment, sustainability. Quality, safety, and to put it from that perspective, it is sustainability. What do you mean by sustain? Any idea? What do you mean by the term sustain in English? It is to nourish. It is to protect. Okay. Why did this need come about sustaining life? What happened? So what the naturalists or the environmentalists, see, we need to give due respect to the education all of us undergo, correct? Some of us do chemistry, some of us do biology, zoology, botany, microbiology, biotechnology. And of course, we have chemical engineers and others, biomedical engineers, etc. Likewise, we have environmentalists, we have environmental scientists, we have naturalists. We need to respect them. We need to respect their research. We need to respect their science because they are coming in from a very different perspective. What did they start noticing? This was about 2 decades ago, a little more than that, where they started noticing that this particular, the universe that we all live in, there were changes, climatic changes that were happening. What do you mean by climatic changes? There were changes that, for example, there were glaciers that were melting. Those days, earlier days, the glaciers never melted. I mean, they noticed it probably at a rapid rate than what it should. You know, you and me realize in our hometowns, my hometown is Bangalore, Mysore. We notice that in these, the rain quality differs, right? Even in your hometowns, you would have noticed this time the rain didn't come much, or it came more, or it's too much of heat, etc., etc. Likewise, the naturalists, the environmentalists study the entire earth and, of course, their area, the flora and fauna. The flora and fauna will give a lot of indication as to what is actually happening to the soil and to the environment. Okay, so that is one thing. They notice glaciers were melting. Secondly, they noticed that the oceans, if you see the seashore, there is a certain seashore that is there. The seashore started receding. That means the ocean started expanding. Why do oceans expand? Because of the heat, because of the temperature. You know, you and I have studied in our science that solutions expand, right? The molecules undergo expansion because of heat, thermal. Okay, because of that, the seashores have started receding. Okay, then they also started noticing a pattern. What pattern did they notice? They noticed that the migration of birds and the animals, the sea animals, the birds, etc., were getting affected. They were noticing certain patterns. Okay, so this is where the environmentalists started cautioning the government, saying that, listen, things are not going okay. We need to do something about it. In the meanwhile, they also found out that the ozone layer was thinning. What is ozone layer? What is ozone layer?
It's a protective layer, man, at that
It is the
atmosphere.
protective layer, atmosphere, the stratosphere. In that, one of that is the ozone layer. And that was protecting our earth from harmful rays from the sun. They started noticing certain pockets in those layers, wherein, you know, the direct the UV lights were hitting. So they also realized that it was thinning, and they realized that it was being caused by CFCs. Any idea what it means?
Chlorofluorocarbons
Yes, chlorofluorocarbons, CFCs. Where were they found?
in refrigerants, man, mostly.
Pardon?
Refrigerants.
Refrigerants, in compressors, in those aerosols. Like, for example, you have these miniature fire extinguishers. Okay, and that's where they found out it was in what do you call, refrigerators as refrigerants, etc., etc. So considering that, the world, the governments of the world, just like how it has happened with other ICH and the other UN bodies, the ISO bodies, the world came together and they said that no, we need to somehow reduce this entire pollution that's happening. So that's the reason why they all met in probably in the 70s or the 80s. They met at Rio de Janeiro. Where is that?
Brazil.
Brazil. So that's where they met, and they decided that they are going to pledge themselves to protecting the world. That's the reason why it's known as the Rio Declaration. It's known as the Rio Declaration, and that's where they all committed. In fact, if you were to go and read on the net, you will notice that it starts with we the people of the earth. That means it didn't say we the people of India, we the people of Europe, or etc., etc. It said we the people of the earth or the universe. That's how the whole thing starts. Okay, that's where they put in checks and balances. They all pledged, and that's the reason why the carbon footprint, I'm sure you are hearing that a lot in the papers everywhere. That's where the governments across the globe started looking at carbon credits. In fact, there are carbon credit audits that happen. Okay, so then what happened? They decided to ban CFCs. What happens when you suddenly ban these things? You can't just ban them overnight. They give a timeline in which you have to sort of withdraw from that particular chemical. So what did they do? They went back to their drawing board. That means they went back to R&D. They relooked at the life cycle of the product. That's where the life cycle thought process of the product came. That means cradle from the beginning to grave. That means it's final discontinuation or withdrawal or discarding or destruction. So cradle to grave concept came only then. The thoughts came in then, wherein they relooked at the entire sequence of activities in the refrigerants and the etc., wherever in the various parts of the refrigerator and other what do you call goods. Then they relooked at it and redesigned those. What you and me are seeing today is the evolution of the past 2.5, 3 decades. Okay, so they started relooking at that. And when they started relooking at that, of course, they had to tweak it. In the meanwhile, as they were doing the R&D, you could say, simultaneously, the controls were coming put forth by the government. What controls did it come? Of course, we always had a pollution control board. Across the globe, there is pollution control boards. We have it in India. We have CPCB, that is Central Pollution Control Boards. In the state, we have the State Pollution Control Board. In Karnataka, it is Karnataka State Pollution Control Board. Okay, so all these came up and they decided to zone the different industry. We as pharma or chemicals, we come under the red or orange category. That means we are high risk because we are generating chemicals, we are generating waste, which are hazardous. Okay, we come under that. And as per the Pollution Control Board, there are certain norms we have to follow. Okay, based on which they will give us approvals. Now, coming back to the R&D, they did the R&D, and that's the reason why you find fantastic refrigerators without, these CFCs in it. And also, the controls also came in the cars, in the vehicles. What you see on the cars, Bharat 4, Bharat 6, what is that? Any idea?
Registration.
Bharat standards, what is it? Any idea?
Bharat for India, right? Single registration, right?
Sorry, you were saying something, Irene?
It's like a single registration for all. Is it that?
Not only that, it is basically that's
control
the
emission,
different.
ma'am.
What do you call? What you're talking of registration is different. This is coming for the car itself. That means emissions testing standards. So Bharat series of standards, they come under the again emission testing standards. So for example, if I'm having a car that's about 15 years old, it may be as per Bharat 2 example. Now, if I were to buy that car, because they are already sending up process that cars which are 15 years and older need not be on the roads anymore. Earlier, they used to do a fitness certificate, FC, and they used to use it. But nowadays, they're asking us to do away with those cars. Okay, so we will have to move on to a new car, which of course will have, say, Bharat 4 or Bharat 5 or whatever the current Bharat standard is. That means it's the emission testing standard. That means whatever is getting emitted is within the limit of that Bharat 4 standards, just like our specifications. In our QC testing, same way, specifications. So this is how the countries of the world, the different bodies of the world are putting controls. Also, when the Pollution Control Board is giving us permission to use, to set up an industry, they say a certain percentage of the factory, I think 30% if I'm not mistaken, or 20%, or probably 20 to 30, should be green cover. That means it should have green cover. That means gardens, plants, etc., etc. Okay, and of course, the design of the buildings, many of the buildings nowadays is also environment friendly. Okay, now why is Bangalore facing crisis? That's another matter. That the kind of landscape that has undergone changes, like it used to be so green earlier with so many fields are now having only buildings and construction and glass. Okay, that's generating a lot of heat. And also, of course, the monsoon is also played havoc this time. Anyway, now coming back to the industry. So sometime in 2002, 2003, I got an opportunity wherein I was requested by Ms. Kiran Mazumdar Shah to implement environment health safety program for the Biocon group. In those days, I think we were 1,500, 2,000 employees strong. And I was wondering whether I should, take it up because I belong to quality and EHS was something I'm not used to. Again, I probably didn't want to come out of the comfort zone. You're very safe in your area. But then I thought over it, had a discussion with her, and then she said, no, you need to implement it for the company. And you would be doing it. And please treat EHS department as any other department, and quality needs to lead it. So probably first time I got an opportunity to implement environmental safety. And I had to study it, just like how you are studying SMF now. Like that, every other day I was studying it and also side by side implementing it. So please remember, just sharing my thoughts, because the slide deck is something that I can just show and breeze you through. Please remember, when you are implementing anything or when you're leading, because we are looking at you as leaders. Tomorrow, when you enter the industry with your background of UG, PG, or engineering, whatever you have learned, we are looking at you as leaders. We are looking at you as solution providers. Okay? You need to ramp up your skills. You need to understand that environment, health, safety, or sustainability, as we call it as, is all part of the work that you are doing. It needs a sort of cultural change because we are so used to doing testing, for example. But I'm giving you an example of what the culture was earlier days. We used to do testing. And probably not all of them were discarding the stuff properly. So we had to bring in a certain systems into place. We had to train people. We had to relook at the SOPs. Okay, so that's the reason why we needed subject matter experts, SMEs. So what we did was we created something known as EHS core team, I think, which exists even today. We created something known as EHS core team, that is the head of the department and the, representative from the lowest cadre of the designations and the mid-level managers. Of course, we had representatives from different, cadre in the designations in the department. They all became part of EHS. Why? Because we needed inputs from them. Any program, please remember, as a department head, when you become one, please remember it is always collaborative approach that sort of enhances the company's growth and your growth also. They look at you as leader because they start approaching you for troubleshooting. They start approaching you for problem solving. If you're not approachable, if you're so focused on yourself, then it's going to be a detrimental aspect to you. So considering that we created, I'm talking of a time, you're looking at Biocon, if you've been to Biocon and Syngene Biocon Park, you're looking at everything set up there. You have signboards. Have you seen the signboards? Have you seen the security? Have you seen the badges you need to wear? Have you seen the emergency layouts? Have you seen the safe assembly point? All this did not exist those days. So we had to literally do it from scratch. And it was a very, very interesting, very, very, learning phase of my career, because I learned everything from scratch. There was no class like you where they taught us environment management system. Of course, there were lead auditor course, but there was nothing running during the time I was implementing. In fact, I became a lead auditor after I faced the inspection. I underwent the lead auditor environment management system course. So always remember, environment, health, and safety go together along with quality. So we had to create all the, the fire maps, the fire protection. I'll go through those things with you. The fire protection, that means get the people trained on fire extinguishers, fire hydrants. Again, you know, in fire extinguishers, we have variety, the dry chemical powder, the foam. So many varieties are there. Then, of course, we had to get people trained on first aid. Then we had to get people trained on fire hydrants. Then, of course, we had to get the pest control teams. Then we had to get the, fire protection service provider who come and regularly, I don't know if you have seen, go out and see. In the fire extinguisher, there are, service maintenance cards that are there. Just like how we have for equipment, there are fire extinguisher numbers. It's all mapped, and then there are cards over there. And of course, the fire extinguisher or the service provider for fire protection, they come and service every other day. They in fact check the weight of the fire extinguisher. Why? To know whether it's depleted or if it's functioning fine. Because when there is a fire, obviously the fire extinguishers have to work. You can't keep sitting and banging it when there is a raging fire over there. Okay, likewise, we have fire detectors. We have smoke. I don't know if you have seen smoke detectors. I think it's there above you. I can see in some areas. Okay, then in some areas we have sprinklers. In some areas we don't have sprinklers. Instead of that, we have dry chemical powder. Where do you think it should be there? Dry chemical powder. Dry chemical powder.
No.
Electrical connections. You can't use water there. Correct? Okay. Next, where do you think we should have fire suppression systems? You need to understand the science behind it, because you're so used to reading our textbooks in terms of science, but how actual application we need to think. Why do we need to, or rather, why does fire burn? Why does it burn even brightly?
Excess oxygen, ma'am.
Excess oxygen. So you're trying to shut down the source of oxygen, right? So you're going to use the various mechanisms. So in case of fire suppression system, where is it available? Where do you think we should put it? Okay, let me tell you what fire suppression system does. Fire suppression system, it sort of, you could say, alerts first. There is 1 or 2 alarm. Again, it's set by the company. And it gets activated. Okay, the 1 alarm rings and it gives a couple of seconds so that people can evacuate that place. And then the gas is released so that the fire is suppressed. It suppresses the oxygen in the room. Okay, so you have fire, you have fire suppression that will blanket the end. After the first alarm, second alarm, it blankets the entire area. Where do you think this should be used? In archives, in documentation area, and in data centers. Why? Because archives have documents, correct? We're storing all our documents for the period of 5 years, 10 years, 20 years, whatever the periodicity is based on the standard. We are storing it there, and that's why, though we say we have IT enabled in many cases, but we do have batch records and other documents which are stored there. Which is the other area? Data centers. Which are the other, again, data center, why? Data center is, you could say, the heartbeat of the organization. It is the network. It is like the, brain, you could say, cortex. Why? Because the entire data goes and sits there. It sits in those, what do you say? What do you call that?
Servers, ma'am.
Yeah,
Servers.
in those, either it is cloud server or it's in hard disk backup server. It sits there. So these data centers need to be protected. Again, we can't have water. It is fire suppression. Okay, and in data center core area where the actual data center stands, or in the archive where the files are stored, in those two areas, people do not sit. People do not sit. Okay, they do not sit. Because why? Because that's not the norm. They sit in their offices, just like how we don't sit in the lab. We have a laboratory office area. That's where those people also sit. So this is how companies have been addressing the requirements. And also what we told the departments to do is to identify the potential source of hazard. So we did a very important exercise in both environment and health. So it's known as aspect impact. You can note it down because I've not included in the standard. There is something known as aspect and impact, hazard and risk. Okay, what do you mean by aspect and impact? Cause and effect. Aspect is the one that does the cause, and hazard is the one aspect and impact. Aspect is the cause, impact, cause and effect. Impact is the effect of it. Likewise, risk is the cause, hazard is the effect. Let me give you an example. Very simple example, which we see day in, day out in our work area, in your work area also. You have people, the housekeeping team, mopping the floor, right? Very neatly, they would have put a stand over there saying, wet, slippery floor. Do not walk or run. What do? We have to walk during that time, and we walk so fast that the hazard is this. The hazard is falling, right? The hazard itself is the wet floor. The hazard is the wet floor. And you walking fast, slipping, hurting yourself, yeah? Or not hurting yourself, but regaining your balance. So you slipping and falling down is an accident. You slipping but regaining your balance is known as an incident. Okay. So this is known as aspect and impact, hazard and risk. So what we ask the employees to do across their area is, for example, it could be the location of their office or the chair. It may not be placed appropriately. It could be the location of the lab. It could be the location of the equipment, instrument. Everything was considered, and they identified which are the areas which can identify the hazard and risk and aspect and impact, and we made a register out of that. That means, for example, if you're handling a chemical, that itself is a hazard, right? And again, you have the risk analysis calculation. Remember risk analysis? Do you remember risk? I don't see everybody nodding your head. Do you remember risk assessment? Yes, you remember risk assessment.
Yes.
On similar lines, the aspect impact, hazard and risk analysis was done. That means every activity for example, if you are doing an experiment, what are the chemicals you're using? That means if you're using acids, bases, solvents, how are you handling it? Okay, one of you said in the beginning of our session that the chemicals have to be decontaminated before it is released into the effluent systems. So we asked the departments to identify the various effluents they were generating in the department. I don't know if you have seen in the labs, every HPLC, mobile phase is kept in a secondary container. Okay. And also the underground tanks that is used in the solvent yards, that is used in the, say, water area or to store any chemicals, all underground tanks had to be having a special kind of construction, which does not release the chemical into the soil. Otherwise, the soil is going to get contaminated. I don't know if you have seen the petroleum installations in the facility. If you were to see that, you will notice that there is something, any truck with the solvents or a petroleum chemical coming into the company, it undergoes a check. What kind of check is that? It undergoes a check wherein they find out if there are adequate brakes. Then they find out, of course, the papers. Then they find out if the people who are there is above 18 years, because as per the law, Factories Act, we cannot allow anybody who's under 18 inside the factory. Okay. So they check all those things, and also they give them something known as spark arresters. So when they go to the unloading tank, okay, they earth the truck. They just don't take the hose and just start pouring it into the underground tank or to the overhead tank. Because you and I know there are a lot of charges there, correct? There are a lot of electrical charges which can create a spark and which can blow up. So considering all that, what they do is they earth the tankers, okay, and they also have spark arresters that are put in there, and then only they start downloading or unloading the material into the underground tankers. Once the tank activity is all done, then of course they put it back. So what they do is they also ensure that wherever the dump storage yard is, there is adequate concrete over there. Why? It is not directly kept on the mud or on the soil. There is a layer of concrete on that, and then only they keep the drums. Why again? Because if there is a leak, chemical leak, it does not go into the soil. Okay, that is about chemicals. Now, in the labs, what do they do related to the chemicals that is being used? You and I use. You and I are scientists. We are, say, for example, if we are chemists, we should know what category, what type of chemicals are being used. What are they? So, for examples, there is something you can go and check on the net. There is something known as compatibility metrics. How can you handle which chemical can be mixed with which category of chemical? Like, ketones, acids, bases, solvents. So there are very, very clear distinction. So this particular compatibility metrics is something that employees are aware of, and they also know how to handle it. And of course, we have the MSDS, if you remember, materials safety data sheets, and those safety data sheets and PPEs are worn, and they handle those chemicals for the reaction. And now, after the reaction is done, what do they do with the chemicals? Do they just pour it into the sink? Should they do that? Excuse me. They should quench it. That means they should neutralize that particular reaction. Of course, get the material out, precipitate it out, then neutralize it, and then only there are again bins in the labs, and they are supposed to put it in those bins only. They cannot repeat, they cannot put it in the sink because it can be detrimental to the environment. So now this is about chemistry. Can anyone tell about the biology aspect of it? Any thoughts on the biology aspects of it? I want you to discuss on the biology aspect of it. I'll give you about 10 minutes. It's now 9:36. So at about 9, I just want you to be online. I'll be mute. I want you to find out the what do you call biological aspects of it. How are they discarding stuff? How should they discard the stuff? What happens to it? Please start discussing among yourself. 9:37 now. 9:47 we come back. Please discuss among yourself and how do we handle the material in the biologics. Anyone ready? Shall we start, please? Somebody like to start?
Ma'am,
Yeah.
so there is a various disposal methods.
I think you need to take the mic.
Yeah, it's
now
here.
please speak. Yeah.
Hello.
Yeah.
Okay. First, the very first method, what I saw is a returning of that things to the manufacturer or donor. One method.
Mhm.
Then, and coming to the disposal method, it is for one is high temperature incineration.
Okay.
Okay. Then, like landfill,
Okay.
landfilling the products. Then burning in an open containers,
Okay.
chemical decomposition,
Okay.
then uh, open uncontrolled non-engineered dump. I don't understand what is that.
Okay.
Then fast flowing water goes like that.
Anybody else has any other thoughts before I start speaking? So there are two things that gets used during biotech or biologics manufacturing. One is chemical, the other is microbiology or mammalian cells. So how do you handle that? If you notice across the manufacturing area, there are signboards, of course. Secondly, if you notice the fixtures that are used are all flame proof. And in many areas, it is fire retardant cabinets, especially in archive areas. Now, coming to the hardcore manufacturing, they are going to use chemicals, and you know how they handle the chemicals. Now, coming to the broth that has the strain, once the upstream is done, the downstream activity gets initiated. What happens to the retentate after the extraction is done? What happens to the entire mass? Those are cells, right? Those are huge volume of cells, huge quantity of cells. What are they going to do with it? One, they're going to test whether there are any, of course, it has to undergo the, denaturation process. Like that means killing, as we call it, as wherein it's exposed to high temperature or differential pH. And that's when they sort of gets inactive, mostly inactivation step, they call it also. So what happens during that time is they will, what do you say? They will take the strain after the deactivation process. The microbiologists check whether those strains are still active or not. All this would have been verified during which process? Which process? Cleaning validation and process validation. Remember? I hope you remember cleaning validation and process validation. They will verify the strains, how it is going to be handled and all that stuff. Okay. And then they will, what do you say, handle it appropriately. So then how does that material go? That material goes into the effluent stream. That effluent stream, what happens to that is, again, it is segregated based on the total dissolved solids. You know that there are ETP, effluent treatment plants, in the industry, in the utility area. If you go in the back of, in the rear of the Biocon Park, you will notice that the utilities, boilers, everything is there. Over there also is the effluent treatment plant. That's where they handle these different streams of chemicals and other material coming in. Okay. Then, of course, these solids, you just can't throw them anywhere. No landfills, nothing. There's something known as multiple effective evaporator, MEE, it's called as. Please look up the mechanism of that. Okay. MEE is something that they use based on the TDS. Accordingly, it gets sort of processed, and what gets generated is the ash. And that ash, again, day to day, they're supposed to store it in the sack and label it accordingly. And also there is a area where they store it before it goes to the what do you call municipality, wherever they get the pollution control board tell them to finally discard it. So this is how companies handle it. Remember, you cannot allow a live strain into the effluent treatment plant. It has to be deactivated or inactivated and then only released into the ETP. Okay, these are the things you need to keep in mind, especially because you are going to handle various activities. What happens to food waste? Again, food waste goes in, many companies now have their own, what do you call, area where they process food waste, like how we do at home. They do vermicomposting, and that is used in the garden. What happens to wastewater? A lot of water gets generated. What happens to that wastewater? Again, water again is not discarded. Of course, there's rainwater harvesting and other things. In the industry, again, there is, as you know, there are different types of water, processed water, and etc., etc. What happens to the water that is coming out of these various aspects, even from the sanitary area and everything? That gets treated. There is a treatment plant, water treatment plant. Please remember that it is different from water system. That is different. This is different. This is wastewater treatment plant, wherein it has softeners. Of course, it has filters, etc., etc. And it goes through the series and they verify the whole aspect of it. And typically those treated water, that's what we call it as, treated water is typically used in the garden area. Okay. Again, different companies have different policies. Accordingly, they will handle it. So this is how companies manage environment, health, and safety. Another thing I want to tell you is how do we handle labs which other than do testing of animal or human samples, right? In many companies, those labs are also there. So how do they handle it? It comes under biomedical waste management rules. Biomedical. Remember this thumb rule, say for example, you cut your hand, you wiped it with cotton. This cotton is contaminated. I know you will say it's my blood, but no, it's contaminated cotton. It has to go there. Again, there are bins there, color-coded bins. Everywhere in the industry, there are color-coded bins. Okay, for dry waste, wet waste, electronic waste. Now, coming to what do you call, I was talking about biomedical waste. Again, there are bins, red color, yellow color, blue color, and all that. So again, government authorized people will come and collect it, take it. You have to account it for. You have to give the number of quantity. What is the kg of waste you have collected? They will also account it. They will take it to their respective areas, authorized areas, and they will incinerate it. Like for example, you may be asked, how do they handle expired drugs? What happens to them? It can't be consumed by humans. So what happens to them? It has to be handled as per the biomedical waste. You need to bring it to the what do you call, company, account it for, then send it to the incineration. Okay, that is one thing. That is about biomedical. We spoke about chemical. Again, in chemical industry, we have to handle it as per the hazardous waste management rules. Okay. Then we have radiation waste, isotopes that we use. Again, it can't be discarded. It has to, again, you have to ensure that the half-life is done, and then only you have to, again, there is a certain protocol they have to follow as per BARC, as per AERB, Atomic Energy Regulatory Body. According to that, they will have to discard that waste. Okay, so this is how the industry, the chemical industry or the pharma industry handles waste. Now, coming to another aspect is, again, as you know, in environmental aspects, you have various pollutions. Now, I'm specifically speaking about the hazardous waste. And as per the law also, and as per environment management system, which we'll go through in my next class after this break, is that we need to have a legal register. What is this legal register? This legal register is the register of all the laws that a particular company follows. Environmental laws, quality laws, that means drug and cosmetic. Then we have the finance laws, then we have HR laws, labor laws, then we have IT laws. Okay, so there are a lot of laws that we all have to follow. And those laws have to be adhered to. That's what I said. Laws are there. We have to address those requirements. Like, for example, boiler inspectorate. Remember, we discussed boiler inspectorate. We discussed what do you call electrical inspectorate. So all these people will come in for inspection. So what we do is, just like how we have been reading about our 21 CFR Part 2, 10, 2, 11, ICHQ7, GMPs for sterile products, etc., etc. We have to break down that law into different, line items and see who's responsible for it and how will it be addressed. Once the law is declared, everybody in all the departments, that's why we have EHS core team, all of them will get alerted that the law has changed and they will have meetings, they'll have gap analysis to address that particular requirement. And of course, we have signages across the organization. So how we manage this, whatever aspect that we've discussed till now, how we manage it is the crux of environment management system. Okay, so I'll give you a break now. We will come back at 10:15. Okay, and get on with the particular slides. Okay, and you can ask your questions then as well.
Okay, ma'am.
Yeah, thank you very much.
Thank you.
Thank you, ma'am.
So, considering our discussion earlier, what is EMS? I'm going to use acronyms now. Very importantly, it's a method and procedure for aligning corporate strategy, policy, operations with principles that protect the ecosystems. So we all know what we need to do across the organizations, across the industry. And also to control human impact on and interaction with the environment in order to preserve natural resources. So in many of the areas, you will see they adopt lakes, like the way the Biocon group has, wherein they sort of, protect the environment. And that's how they address the requirements as well. And of course, adhering to the International Organization for Standardization, that is the ISO 14001. So just to give you a little background, the ISO series, as you know, was initiated sometime in 1947. It's based in Geneva, Switzerland, and it has thousands of standards. You saw just now, 14,000 series is for environment management system. Like 9,000 series is for quality management systems. Not that they consciously asked for that number, but it so happened that they got those numbers. Now, also there are standards for environmental monitoring. Please remember, environmental monitoring is different from environment management systems. Okay, air quality and all that, it's a different thought process. So that is one aspect. The next aspect is ISO 14001 is, if you look at it from the perspective of the international standard, it is actually a voluntary standard. Okay, and but many governments have adopted that and they have put that as a requirement in their pollution control boards. So now it's part of the pollution control board's mandatory requirement, wherein they ask the company to have a ISO 14001 in place. Why? So that they address those particular aspects in the industry. And that's why all the companies, whoever is generating waste, will have to adhere to the 14001 standard. So now companies do not only do environment standard, EMS, they also do something known as health and safety standards. So that's why it is called as EHS. These two go in combination, environment, health, and safety. So this health and safety standard is basically an occupational health and safety standard. And that is the standard, that standard number is ISO 45000. Okay. So what do companies do? Because these two standards, I have studied it, I have implemented it many times over. So these two standards are similar. One speaks, the thought process, everything is similar. One speaks on environment, the other speaks on health and safety. Since both of them go together, because we are handling chemicals. When we are handling chemicals, we have to wear PPE, etc., etc. And when we're using chemicals, we have to discard them right, correct? So it's more or less, it's linked together. And that's why across the industry, it is environment, health and safety standard, or some of them call it as SHE, that is safety, health and environment. Some call it as environment, health, safety. Then some call it as health, safety and environment. So it varies in the industry. They call it in different acronyms based on the way they handle that particular activity in the organization. So what does this look at? The whole environmental management, what does it talk about? It talks about, let's go one by one, ecology. Like any company, before they set up the, factory in a premises, like if you're buying a land and we're going to set up an industry, the first thing, of course, is we have to get a clearance from the Ministry of Environment and Forest, okay, or the Pollution Control Board. Pollution Control Board comes under MOEF, that is Ministry of Environment and Forest. So we have to take the permission from them. So before that, they want us to do a survey of the flora and the fauna, the soil, everything they want us to do. Okay, and then only they will agree for that particular area. The government, before they decide which area for the industry, they would have done all this also before handing it over to the industry, and industry also should do it. Once that is done, of course, they are looking at the earth science, then they're looking at the atmospheric science, then of course, the water related aspect of it. And now coming to from a bigger picture, of course, there's a lot of climatic change. For example, if we live in an area that is prone to earthquakes, how is the company going to handle it? So that they do all this activity in the Environmental Impact Assessment, EIA. That is something that includes all these parameters, especially until the environmental assessment, that entire from green to this below on the left side, you can see. So what they do is they do an environmental impact assessment to assess what are the activities being done by the organization, proposed activities, because you still not have built it. And they, of course, take into consideration the monsoon, the climate, the water source, of course, what are the atmospheric requirements that is needed, if it's too hot, those kind of things. And then, of course, the soil also is reviewed. So this entire thing will be reviewed and it would be coming out in something known as environmental impact assessment, which the Pollution Control Board will review. And after that only they will approve, or they may ask additional requirements and then they approve the company. Now, as I said, environmental law, we need to know what are the laws that impact our industry. If I build a company, what are the laws I need to consider? How will I consider? What should I do? Like, for example, emission testing is also there. If you notice in the area, they have a lot of scrubbers. So what are those scrubbers? They ensure that the harmful elements do not go into the air. Okay, they have different micron scrubbers. They have different exhaust types. So all these are taken into consideration by the engineering team when they are involved in the facility design. And also the projects team, the people who actually construct, they also get involved, and that's how they decide to build the building. And also, as I said, they have to adhere to the law. They'll again map all the law requirements, legal requirements, and they take it appropriately. Now, coming to after environmental assessment. So now, you and I know that in the various departments, they're generating different types of waste. So how are you going to monitor and handle it, manage it? Mitigate means managing it. How will you manage it? You could say these are sort of case studies for us to discuss from our industry. How will you handle? Say, for example, we need to handle, for whatever reason, the company has decided to set up a lab that's into, say, for example, high risk testing. What is high risk testing? One is viral testing, because we are handling biologics material. We have to do viral clearance, yes or no? Viral
Yes, ma'am.
clearance testing
ma'am.
is important. So,
Yes,
but,
ma'am.
and you need a virology lab for it. And if you need a virology lab, you can't have an ordinary lab. You need a separate lab. Okay, then we'll come to that. Next, if in case you're setting up one with isotopes, that means radiology. Okay, then you need to, in the chemistry area, you're going to handle cyanide reactions, high risk hazardous reactions, HPAPI. How are you going to handle this? Any idea? Any thoughts? How will you monitor it? How will you measure it? You will have to handle psychotropic drugs. How will you handle that? Again, as you know, we need permissions. Okay. And of course, we need to have procedures in place. Again, we would have done aspect, impact, hazard, and risk analysis. Identify which are the cause and effect, how you're going to control it. For example, let's take isotope, because isotopes are used in biological research. Okay, more importantly, during the various genetic engineering manipulations, they use it in small bio labs. So how do these companies handle it? So they follow as per AERB, Atomic Energy Regulatory Body, that's based in BARC. Now, again, you need the necessary permissions, and of course, you just can't do it in an ordinary lab. It's a special kind of lab. The specification is given by the AERB, and we have to construct that lab as per that. And it is also to be handled by a person who is very skillful and knowledgeable in it. Okay, it's not typically not a fresher. It is somebody who has got experience. So now, once they prepare the layout, get the approval of the layout, then they construct it. Once they construct it, they come in for an inspection. And of course, the employees, whoever are going to be there, are also trained. There is a radiation safety officer also who is there. Okay, and that person also needs to be trained. So all these people are trained very well by the AERB. And then, of course, they're given special badges. So what are these badges? These are known as dosimetry badges. Basically, whoever is going to work, that's the requirement as per the law. Whoever is going to, you could say, work in the lab area should wear this badge and then only work. They cannot ignore that badge. What is so special about the badge other than your identity card? So typically in those areas, they wear that particular badge and then they do the various activities. And after that activity is done, of course, as I said, the material is discarded. It's not disposed of into the ETP. They wait for a half shelf life, half life of the product of that particular isotope, and then only they discard it accordingly. So now what does this badge do? Once every 3 months or once every alternate month, this badge needs to be sent to a government approved laboratory where they check if you are exposed to any radiation. They verify whether you are exposed to any variation. So if you are exposed, then automatically that badge will detect it. That is the importance of that particular badge. And again, discarding, they have certain, poly bags, which are thick, which are sort of tagged, which are numbered, what quantity was disposed, everything is accountable. That means every microgram, nanogram, whatever quantity you're using, everything is sort of monitored. This is known as monitoring. And also the badge is also known as monitoring because you're sending the badge to verify if you are exposed to radiations. So that is known as monitoring. If in case there is any such kind of a incident, then immediately they'll have to take action. Correct? So that's how industry do it. Now, that is about the lab. Next, we go on to the viral testing lab. In the viral testing lab, again, it is high risk area because they are using live virus, not some ordinary strain. This is a live virus. So that's the reason why they need clearances. And that's the reason why, again, the layout and all is checked. And when they release the, license, when they get that virus, again, they have a cell banking system, if you remember. They do it through that. And of course, those are stored very, very in a controlled manner under lock and key. Not all of them have the access to open that particular, vial or whatever it is. Again, they wear special suits and then only they work in isolators if it is required. Now, let's get on to, from lab, we move on to the manufacturing area. Now, again, in the manufacturing area, different areas where the waste is collected, like for example, wastewater automatically goes to wastewater treatment plant. Okay, like that, they will have designated, areas and process how they handle it. Now, in spite of all the precautions you and I take, by chance, if there is a spill, what are you going to do? If there is a spill in the area, what are you going to do? Okay, supposing we are bringing a drum. Okay, the pipeline didn't work, so we're bringing the solvent in the drum to the manufacturing area. If there is a spill, what are you going to do?
Report to the supervisor.
Okay. Supposing you are the supervisor. So when you are carrying those containers, as if you remember, we said secondary containers should be there. Okay, you will see in the labs that there is a separate cabinet for acids, separate for solvents, separate for bases. Okay, it all has an exhaust system. Number one. Number two, when you are carrying the particular chemical into the manufacturing area, it is always under a secondary container. That means if you are taking the container, you are having a secondary container, and this is what is going into the manufacturing area. It just does not go like this. Okay, typically solvents go through pipelines, but I'm giving an example. Okay, so that's how they handle it, secondary containers. Even if you notice, you may have noticed when you're going around in Biocon Park, they'll be carrying certain ice box, or they'll be carrying a sampling, bag or a sampling kit. It's all within that so that, it doesn't spill out. These are all the precautions. And also when they are carrying, sample from, say, I'm carrying the sample from R&D to analytical lab, I need to carry it in a beaker, again, a secondary container. I'm keeping the the test tube or the capillary tube and inside a particular beaker, and then only the beaker probably in some other carry bag or a carry storage container, and then I'm taking it. I'm not taking it like this. So please understand that there are differences. Now, what happens when there are electrical charges in the product you're manufacturing? So the companies have flame retardant fixtures, correct? And they ensure that when you and me walk into the manufacturing area, we wear safety shoes. The safety shoes, if you notice people wearing in the manufacturing area, those are anti-static shoes. That means there's no static there. And even when we enter, there are pads that are given. I don't know if you have seen. Next to the entry into the manufacturing area, I've seen it more in the industry where they manufacture chemical products. Because the charges are more here, you could say because of the biological aspect, it may be less. But whatever said and done, there are huge pads, probably as big as your screen monitor, or bigger than that, which are placed outside the room, which we need to press our hand to it for about 5 to 10 seconds. The instruction is very clearly given there, saying 5 seconds, and you count to 5 or 10, and the charges dissipate into that particular anti-static. And then you enter the facility. Again, you are ensuring that you are safe, and also so is the environment. So I'm not going to discuss about environmental monitoring here because environmental monitoring is a separate topic. Okay, so now in continuation with this other type of environmental monitoring, what do I mean by that? So when they are using chemicals, especially chemicals, and especially solvents, you and I know solvents evaporate. Can you give me examples of solvents?
Benzene.
I can't hear you, Irene.
Benzene. Benzene.
Benzene.
Either.
Okay. Our favorite acetone.
Ethanol also, ma'am.
Yeah, our favorite alcohol, our favorite acetonitrile, methanol, all our favorites. Why am I saying it's favorite? Because it's used more often. It's like, it's a routine thing that they use. So otherwise, please don't take it otherwise. It's not for drinking consumption. So these are the chemicals that are used typically, and there's a lot of fumes that are generated, which you and I cannot smell. We do wear masks, but also there are certain permissible limits again. So what do teams do? So we have EHS department who have, if I'm not mistaken, the name of that particular machine is odometer, wherein it's a very, very expensive one, by the way. it has got a sensor, and it can, again, it depends on the model. Based on the model, it can detect up to anywhere between 1 solvent to about 10 solvents or even more. It can detect, say, for example, you are using 2-3 solvents in my area. So they do environmental monitoring periodically to check the, the content of the solvents in the air, the saturated air. Basically, they're testing out whether there are any solvents in the air, and because it is also harmful to us. Okay. This is the kind of monitoring that is done. This again is from the, I'm talking from the perspective, not from environmental monitoring, which we will discuss, but from the environmental monitoring of the chemicals. So, and residues, because during reactions also, you know that there are chances that certain odor is generated. Okay, because of various chemical, various process and all that, there are certain odor that is generated. Okay, again, obnoxious smell that is generated, which can be harmful for humans. And this is where R&D plays a very, very important role. So they do a lot of these monitoring, even in R&D area, especially pilot scale, before the transfer technology happens. So even these considerations also are taken. I want you to understand that EMS is not in isolation of the product manufacturing or quality of the product. It is together. Okay. So all these are considered during the EMS also, and especially during the technology transfer. Why? Because there are something known as process safety engineers. It's a very good, by the way, it's a very good career option. Process safety engineers. These engineers are basically looking at the molecule from the safety perspective. Like during the manufacturing of the system, of the product, you could say, they're looking at it like from the perspective of, is it explosive? You know, is it highly charged? How should it be handled in the manufacturing area? Because you're going to scale up so many times, right? So they consider all these things. Okay, and another important thing that they do consider during this particular thing is how should be the viscosity of the material. This is where process engineers come in, process safety engineers. So, you know, what should be the viscosity of the material? What should be the RPM? How should you add the material? Should you add the material at one shot, or should you add it in a slow manner? So say, for example, you put in about a kg and it sort of rotates, and then you put the next, and then you put the next after it rotates. So this is how they plan everything, and this is what gets carried over from R&D to transfer technology. And this is also to say that even the R&D team, you could say the process engineering team, the process safety teams, even these people also do a lot of aspect, impact, hazard, and risk analysis. Unlike in quality, we're talking only of the quality of the product, correct? You're only talking of R&D, then you're talking of process, then you're talking of QA, QC. If you were to read the environmental management standard, EMS standard probably is one of the only standards that speak of the company, that speaks of the facility. Like, for example, I'm giving an example. If ISO 9001 speaks of quality system in Biocon Academy, that means only in your floor, only your area, and only about the education or the skill that is being provided, the quality of the skill training that is being provided. If 9000 speaks only on that. Whereas, if they have to take an environment management certificate, that is ISO 14001, the entire compound in which your building is, that needs to undergo environmental standards. So if you were to notice that the environmental management standard certificate is given for a facility, it is given for a entire company. So in fact, excuse me, when we were going for Biocon certification, Biocon group, for the first time, we didn't have Biocon Park then. It was still being built in 2004, 2005. It was still being built. Okay. So we went only for the 20th kilometer. That is the main campus that is there on the Hosur Road. There were 3 companies there. One was Biocon Limited. The second one was Syngene, which now is in Biocon Park. The third one was known as Clinigene, that was into clinical trials. They were doing human sample testing. So there were 3 companies within that big campus of 20, 30 acres. So when we went for it, we discussed about it with the management because we need approval from the management. And they also had to review it. So when we discussed about it, they said, yes, the entire 20th kilometer campus is going to go for ISO 14001. So that's the reason why you see the entire campus having that particular management system. And also you may have seen the speed limit across in the various areas. That is as per the factory's rules. Okay, and since company has its own vehicles, there are a couple of vehicles companies will have, not only Biocon, others also. So what happens in those company vehicles is that they will have to follow the Motor Vehicle Act, which we all follow, of course, but again, it comes under the legal requirement of the organization. So this is how the, just to give you an idea as to the entire facility, including the housekeeping people, including the production workers. They are not called as workers, but contract workmen. Everybody has to be taken care of. In fact, we give special training to housekeeping staff. We give special training to contract workmen, work person, so to speak. Why? Because they are doing work for us and they are also handling environmental systems in the sense they are cleaning the vessels or they're using chemicals or whatever it is. They are also involved in it and they also have to follow our requirements. We gave them special training as well. One of the things that you may have seen is the installation, those huge installations of air, Praxair, you may have seen it. Even those also, they also have to be taken care of very well. Any installation that a vendor is doing in your company, that means any person who's working on our behalf or any vendor who is going to use our facility to have an installation, they also have to follow ISO 14001 and of course they have to follow our requirements. Remember that, please. It's a very, very, very important aspect of it. Okay, any questions? So another, when I talk about environment management systems, another one that I want to speak about is cylinders. You may have seen cylinders across the organization. Please note that you cannot carry the cylinders as such. You need to have a cylinder carrier that we have. And you cannot carry it on your shoulder or anything. We have installation, again, we have done it over the years. So whatever installation that was there in the first floor, second floor, those cylinders, we decided to keep it outside and next to the wall of the, say, lab area in the ground floor and give a pipeline instead. Otherwise, they will have to carry the cylinders on top. Secondly, do not kick the cylinder. Okay, we have seen many, not necessary in Biocon, Not in Biocon, definitely, because we had trained them very well. But outside, we can see people who kick the cylinders which are filled. Please understand, those cylinders are like missile. They can just, burst. So if you see anyone doing that, please stop them from doing it. Okay. And again, you have to be aware of the SDS, safety data sheets. That's very, very important. And again, if there is a spill that happens, how do you remove it? In the laboratory, there are something known as spill kits that are available. Typically, it is from the 3M that they purchase, or again, depends. It could be in the chemistry lab, it could be in the biology lab, whatever lab, we have to have spill kits. Okay, and also in the outside area also. If in case it spills on the soil, then that entire area has to be excavated, and of course, they have to do soil testing and accordingly mitigate it. Okay, now coming back to the slide deck. You've already touched about the management practice wherein they train. Of course, we do have internal audits also. Just like quality, we have internal audits. And then the waste is managed. The waste generated is taken into consideration. And of course, it is sustained. Aims of EMS is for compliance and to reduce waste. The standards of 14,000 was established in 96. And the current one that we are all using is in 2015. It follows the PDCA cycle. That means plan, do, check, act cycle. This is the cycle for the quality systems also. So what do you mean actually by planning? That means you plan how you're going to handle the environment management system in the company. Then you sort of have a policy related to it. And then, of course, you plan it in much more minute detail. Then you implement and operate it. And any checks have to be addressed. That means the monitoring system comes here, what I already told you. And then if in case there's anything that is going beyond the required limits, that's when you will act on it. And again, it goes back to the policy and the whole cycle continues. Plan, do, check, act cycle. This is similar to what you are doing right now. You are planning your studies. You're going to implement, you're going to plan out saying you're going to do this in the morning, this in the night, whatever time slots you have scheduled. Then you're going to implement it. That means you're going to implement and operate that plan in action. Then periodically you're going to check the progress of your studies, right? Either through the quiz that you will undergo or through discussions, you'll get back to us. And then if in case there's something that requires a little more tweaking, you're going to act upon it. So this is known as a plan, do, check, act cycle, PDCA cycle. This is given by Deming. So again, in the departments, what do? They identify the environmental aspects. That means, if you remember aspect impact, the air pollutants, the pollutants basically. And who's the most important to the company? That is, of course, the worker and of course also the other stakeholders there. You need to verify whether you are within those laws, which I explained to you. And of course, they will derive an objective and target. I'll speak about it in a minute. And then the management actions to support the objective and targets. For example, if the company says that they will minimize the use of power by, say, 5%, I'm giving an example, 5%, that's what the EHS department with the other department heads decide. They will discuss this with the management. The objective is to minimize the use. The target is detailed. How are you going to reduce it? Which department will do what? Like, for example, if it's a huge working area and you're switching on the lights from across all the area, and then you're going to say that you're going to minimize sometimes they leave the lights on in the meeting rooms, in the, restroom area. So you're going to put in these, these pointers saying that listen, we will switch it off after this time or this so and so and all that, so that we will minimize the overall usage of electricity. So this would be the objective and target is this. And of course, you're trying to reduce the power consumption and in turn enabling the environment. So this is how companies do it. Also, it will become our target. That means you will have the company level target, then you will have department level targets, then it will come to me and you. That means it will be a personal level targets also. Okay, like for example, zero accidents is one of the objectives. Zero environmental accidents. That means you will take care of all the environmental aspects in such a manner that there will be no leak. Okay, and also you'll find out a better system to handle the environmental waste that is being generated. Okay, so whatever you see now, for example, food waste, we are saying vermiculture. Initially, food waste was food waste. It was just going into a dump yard or it was just going as a waste to the municipal. But later on, that became an objective of the EHS department, canteen department, admin department, and all of us. Reduce food waste. So I don't know if you've seen in, I don't know if it's there in your particular building, but if I remember right, in the Biocon Park, they have the, notice over there as to what was the food waste that was generated, what was manufactured, that is the canteen food, what was the quantity that was cooked, what was consumed, and of course, what got wasted. What was, of course, wasted is what you get. The differential amount is the what was consumed. Okay, so that if they say we will reduce it by 10% or 5%, again you're going to educate the employees saying if you do not like it, do not serve yourself that, so that you do not waste it. So that's how the companies, I'm giving you examples from different perspectives and different departments, how they minimize waste. Of course, reduce, reuse, and rework. Okay. The do is, of course, the structure of how we handle the environment management systems. The same, remember the same thought process wherein you have the policy, then you have the SOPs, then you have department SOPs, and then you have the records. So again, EMS documentation and quality documentation would be somewhat similar. Many companies, we have it as the same so that, people do not get too much stressed in handling it. The document control would be on similar lines. Operational control procedures are procedures that handle the operation of very crucial aspects of environment. Like for example, spill control, that's an operational control procedure. Then emergency preparedness response, again, is an operational control procedure. Emergency preparedness response is a huge, management system by itself, sub-management system. So what actually happens in this is they identify the people who are fire extinguisher trained, fire hydrant trained, then of course, self-contained breathing apparatus, that's a separate training they will undergo. They will undergo first aid training, then they will undergo other specific trainings. And of course, they will have an ambulance parked always 24/7. And of course, they do have an occupational health center where you have a doctor and nurse on standby. So these are the various, components of emergency preparedness and response. And also they have an ERT vehicle, emergency response team vehicle, wherein it's dedicated only for that. It's a miniature, you could say, containing activities all related to mitigating emergency situations. So all these are available, and each and every department will have its own emergency trained people personally. So if in case there's a fire, for example, when you are in the lab, how are you going to handle it? So more or less in all the companies, they are training the scientists, the QC technologists, everybody to handle the fire. Because see, if when you and I are doing some reaction, and if there is a fire, by the time we call somebody to come and put out the fire, it is going to take time. So in these cases, what happens if you and I are aware of how to use the fire extinguisher, we can immediately handle it. So this is how they have it. And of course, there are mock drills that are done. We also identify. So how do we, this is an entire huge exercise that we did, wherein we had to identify what is the emergency route. Okay. And of course, where should all the employees stand? We should take a head count of the employees to see if anybody is missing. If anybody is missing, then somebody needs to rush and the emergency ERT team, as we call it as, is already available there, and they are doing the entire checks. They take a head count, they take the head count of the contract people, head count of the visitors. So this is how they sort of, prepare for the emergency. And also another important thing is that another emergency aspect is that there are a lot of mock drills that are done. That means they do sort of a dummy runs just to show that, they are all aware of it. And another important thing is that do not use the lift when there is a fire, because the fire will go through the shaft, through the lift shaft, and it catches fire much faster than how instead of the building or the stairs. So when you are going to the malls, shopping malls, or you're going anywhere, or you're visiting a hotel or wherever it is, just be aware. Just be aware about the these emergency requirements. Safe assembly point, which is the emergency exit. Okay, so please be aware of these things. And what should you do in an emergency if you're so for example, we are all scientists, we are all working in the fume hood, and we are doing various reactions. There is an emergency declared. What should you do if such things happen? What should you do if such things happen?
Ma'am, you should exit through the nearby emergency exit,
Agreed,
ma'am.
agreed. What happens to the material? Your reaction?
Ma'am, we have
There
to
will
neutral.
be, nobody is going to come calling fire, fire. Nobody is going to say that. The alarm will ring first. You will run. Before you run, what should you do?
Ma'am, we have to neutralize.
You have to
After that,
neutralize the reaction that you're handling. So these are operational control procedures that are there in all the departments, especially where they handle the reactions or biologics material. So every department will have its own method of handling this. Next, of course, what do you do if in case there is some incident that happens? That means there is a spill or there's a minor fire. You need to immediately inform your supervisor or EHS. Again, depends on the escalation mechanism. And of course, you will verify it. And then, of course, they will come and do the investigation. Again, typical CAPA will be done. The records are maintained. Monitoring and measurement. What is the other monitoring thing that they do is one is the thumb rule, we go back to calibration. All the monitoring device that we are using in the industry, including those related to environment, health, safety, the equipment especially are also monitored. What are these equipment? These are fume hoods. The fume hoods, they check the air velocity of the fume hoods to see if it is functioning fine, so that the exhaust is taking away the unwanted air. Same thing with the exhaust. So this is known as monitoring and measurement in terms of environmental management system. That means all the areas which are generating or potential for generation of waste or pollution, those are sort of monitored and managed. And of course, we do have EHS audit, like how we, sorry, EMS audit, like how we have the audits for quality, we have the same. And of course, we have a separate management review wherein we discuss about the health of environment and health and safety also. And of course, we find out better ways of improvement things. So let me give you an example. Long ago, I'm talking about more than 2.5 decades ago, when we were using laptops and when we were using desktops in the industry, we didn't have screen guards. Okay, we didn't have, these were screen guards that were used because there were a lot of emissions that were happening. Not necessary emissions when I say it's not like the radiation kind of emission, but yes, it was hindering the eyesight, visual and all that. It was quite inconveniencing the people. So they started putting those anti-glares onto the, it was an additional kind of a clip with a, with a board, sort of an anti-glare, what do you say, rectangle thing that was clipped onto the desktop or to the laptop. Then after that, what happened? As you know, technology is much more advanced. Now you have very, very good anti-glare and the latest technology, crystal technology, whatever you call it, in our screens. Again, this is an outcome of environmental management systems. Okay, of course, it comes at a cost. It doesn't come cheap. So again, we did discuss these in brief and with a couple of case studies like air pollution control, water pollution, waste management, workplace environment health programs, other environmental management programs. These are to ensure cost saving, compliance, pollution prevention, increase efficiency, improve morale, set targets to reduce use of energy, water, and waste. So let's look at one example. Travis Perkins supply building material to the trade for over 200 years, one of the largest suppliers. They provide more than 10,000 products. So they want to be a sustainable business. So the legal requirement is, of course, the laws, the pollution prevention and control regulations, special waste such as toxic material, water resources, etc. Financial pressures. Britain has some of the world's tightest environmental regulation, landfill tax, carbon tax. Okay. So what do companies do? They do have market pressures also. They'll only buy from companies which have ISO 14,000. That means it's understood that the raw material that is coming to you is, of course, having the necessary quality, but also has taken into consideration the 14,000, that is environmental requirements. And customers insist that timber comes from sustainable sources. That means you are taking into consideration about the protection of the environment also. I know they are logging timber. That means they're cutting down trees, but at the same time, how can they sort of, have a sustainable, how are they protecting the environment when they are felling the trees? So this is how it's done. They fix a certain area that needs to be felled. And of course, they have to grow more trees, more than probably double or triple of that. So all these programs are there with the companies. So what did they do? They went through EMS. Why? Because of course it's internationally recognized. What did they do? They had their policy, they implemented a plan, and they made the action list and the responsibilities for that. So environmental policy that was influenced by the legislation and the environmental aspects that they have internally. Then they had the plan That means they had their procedures and the operating procedures, that means control procedures. And then they set the action limits and responsibilities via training and key performance indicators. That means it became part of our performance evaluation. That means companies evaluate us based on our work and also how we contribute to quality, environment, health, and safety, and also sustainability. So coming to their interaction, water use, energy use, product buying, water, what were they discharging the water? Quantity of water, how did they reduce it? The waste management of it, air emissions, land management, vehicle impacts, noise and odor. So they say that they are complying with the legislation at all times. Prevent pollution. Of course, there is certain amount of pollution that gets generated, especially when they are felling it and when they are cutting the trees. A lot of pollution does get generated, and also they are looking at continuous improvement. So some of the environment improvement plan that they use is, which I discussed at length earlier, was electricity use, carbon dioxide emission, vehicle emission, storage and use of oil, diesel, petrol, and gas, discharge to the drains, the water, storage and use of timber treatment materials, and purchase and sale of products, particularly timber. So one of the action lists is observing all the legislation, and you're also simultaneously preventing pollution, like preventive pollution, for example. If they're having old model of a generator, and the new regulation says that, it has to be within the permissible limits, that means a specification is set, and the old generator is unable to, reduce it by so much, then chances are that they have to phase out that generator and bring in the new one. Just like our car, which I told you about Bharat and the requirements of the Motor Vehicle Act and the RTO. So likewise, we will have to align to the regulatory requirement. They will give us a certain time period within which we will have to align. Looking for ways to make conscious improvements. Like, for example, in many companies, they used to use plastic stirrers for coffee and tea. And that became too much. I mean, there's so many of us, and if in case we drink so many times coffee or tea, we'd have to use that plastic stirrer. So that was there for quite some time. And then we took a call and we removed that. And then, of course, we got the wooden stirrers. But then again, wood, you're felling trees. So what did the companies decide? What you see today now? They decided to have a container with water with 2-3 spoons, which can be washed and reused. Remember? Reuse, reduce, rework. So one of the important way companies communicate is through communication, internal and external communication. And how do you, handle waste? How do you display the board, have the requirements? And where do you need to throw the, or rather discard the oil contaminated waste? All these things are again decided. In fact, during environmental audits, the audit actually starts with the backyard, the waste pit, the environmental treatment plant. That's how some of the auditors will audit us. So costs and benefits of EMS through a number of cost reductions. Again, prevent waste is the first thought, reduce it, reuse it, and recover waste also is another thought. So again, environmental system takes time and commitment from the entire organization. It provides ongoing benefits, cost savings, and definitely for a good workplace culture. So 14,000 is a series of standards for environmental management system. I think there's one extra zero there. So what are the components? The policy, planning, which I spoke earlier, implementation and operation, checking and corrective action, and management review. So this is the PDCA cycle, you could say. Policy is a statement about the values of the company, and to perform as per the environmental requirements. And it talks about the EMS framework, commitment to continual improvement, pollution, environmental compliance, and it is typically signed off by the senior management. It is, you could say, a decision-making thought process that considers environmental, social, political, economical, and governance factors. Any questions?
Ma'am, you said that environmental monitoring system is different from the environmental management system, right?
Yeah.
Okay, ma'am. So if we go for case studies for the environmental management system, what are the examples we can take, ma'am?
So environmental management system is again, though it's applicable to both, quality of the air within the manufacturing area.
Okay.
Okay. If you look at it from the quality perspective, we're talking of the sterile manufacturing area. And in those areas, we cannot have microbiological organisms over there, correct?
Yes, ma'am.
We can't have contaminants there. Okay, so that is one thing. Whereas in this kind of environmental management monitoring system, again, as I said, they take around the odometer or whatever. I'm sorry, I hope I'm correct. They take that particular machine around to test what are if there are any, if the air is saturated with solvent molecules. Okay. In the case of the environmental management system monitoring, they're using a machine to collect the air to identify the type of gas that is there or the solvent that is there. In the case of especially the microbiologists who do the testing, they take an air sampler. That air sampler is taken and they test whether there are any contaminants there. I hope I am clear. You're okay with that?
Yes, ma'am.
Yeah. Because I'll be speaking separately on environmental monitoring in the next class, class after that.
Okay.
Okay, I'll give you the break because I think the next class is going to be by another person. So I'll take leave. Have a good day. Are you visiting any facility today?
Yes, ma'am.
Yes,
What
ma'am.
facility?
Same S18.
Okay, fine. So now when you go, you're looking at different perspectives. You're noticing things as we are talking, I suppose, compared to what you were earlier. Earlier, you would have just gone and seen around, but now there's much more focus on what actually is happening. That's really good. Okay, see you. Have a good day.
Thank you, ma'am.
ma'am.
Thank you,
Thank you,
ma'am.
ma'am. Thank you, ma'am. Thank you, ma'am.
Give it that Vishnu. Little of that. No. See, Camila. Type. Bacterial. Typhoid is getting from spoiled food or water. And also how it spread. It won't spread through air and contact. It won't spread through saliva. It will just through waste. asked that. Does it spread through saliva? It does not spread through kissing.
Hello, everyone.
Hello, ma'am.
Hello, ma'am.
Good
Hello,
afternoon, ma'am. Good
ma'am.
afternoon, ma'am.
Good afternoon, ma'am.
Not yet afternoon, still
Good morning, ma'am.
morning. It's okay. So, welcome back. So, how was your classes? Because all this while I haven't seen you people.
It is
Last
good, ma'am.
is good, going on good. Okay. So, I think for the next 1 week, so some faculties from Ramaiah College will also be giving you all a brief insight about this sterile manufacturing process. It's all related to microbiology. And most of it you would have already studied this in school, in your college as well, in your BSc or your MSc, your degree classes, you've studied all this also. Some basics about microbiology, right? Yes, I can see all the heads shaking. Okay, so only 19. What about the other Syngene employees? I don't see them.
Only 2 of them joined, ma'am. Syngene employees, Dr. Basaya and Mani. Balaji, Kumar and Rajshekhar is absent for today. Kumar joined for the first session.
No, he's not here now.
For the first session, he joined.
Now he's not here.
And Prashanti, she went back to PG due to fever.
Oh, okay. All right. No problem. So, I think everyone has already, you know, so you've already gone through the slides, right? You've gone through the slides. So these are just some basic topics that we'll be discussing. And the other topic that was supposed to be done, that will be done by another faculty of ours. Okay, so moving on to this sterile manufacturing, that is microorganism and contamination control. Now, do you think is it possible to have a sterile environment, a completely sterile environment? Is it possible?
No.
Definitely not possible. So, can we do that in reality?
No.
Definitely not. So we cannot have, or it's not at all possible to maintain a sterile manufacturing environment. And in any environment that you work on, we will always have microbial contamination. So this is an inevitable thing. And how these sterile environments are devised or how they are designed. So they are just going to be designed in such a way that there is minimal contamination. So we'll have to wear our safety gears, like we have to wear our sterile gowns. We have to protect, our hands, mouth, hair. We have to cover our hair. I think you've already learned about gowning and all that, no? In aseptic techniques, and you've also visited the facilities also where they've shown you what are the possibilities of these sterile gowning processes and all that. Right? So, how and is it possible to prevent any shedding of these microorganisms? Is it possible? Definitely, it is not possible to prevent it, but we can maintain it, or we have to take extreme measures to prevent the shedding of microorganisms. So how do you think that we can have this microorganism or control? See, it's very difficult to maintain a complete aseptic room. Now, do you understand by this word called as aseptic, right? What do you mean by aseptic? What do you mean by aseptic? Any idea?
Controlled environment.
Controlled environment, or
Where the NVPC and VPC are in a particular limit.
Irene, I didn't get you.
Aseptic means where the NVPC and VPC count in a particular limit.
Okay, in general. Okay, in general, aseptic means it should be free from any sort of contamination. Now, contamination can be caused by either harmful bacteria or any, viruses or mycoplasmas, anything. It can be any type of microorganisms. So when I'm saying, when I referred to this word called as microorganisms, now you know that there are many types of microorganisms, right? Can you tell me a few microorganisms that you know, or a few groups of microorganisms?
Bacteria, fungi, molds, yeast.
Yes. Molds, yeast and all is the same only. It belongs to fungi. Okay. So microorganisms, you have the major classification is bacteria, you have fungi, you have viruses, you have mycoplasmas. So these are all the major, things that cause contamination. So how do you think that this, how can we control them? How can we control these microorganisms?
By controlling the environment.
By controlling the
Environment.
environment. How can you control? Do you think you can control the environment in nature?
By
Definitely.
sanitation.
Yeah, you can take measures to prevent contamination. So basically, what we can do is we can, we can start with any manufacturing. So when you talk about whether it is a pharmaceutical process, see, microbiology is present everywhere. Microorganisms are present everywhere. It's very difficult for us to maintain a complete sterile environment. Now, whenever we talk about any kind of processes, whether it is related to pharmaceutical processes or whether it is related to any other type of industries also, it can be food related industry. That is where food is very important, right? And any kind of equipment manufacturing processes. When you talk about batteries, okay, lithium batteries, it can be any industry. Or when you talk about solar panels also, when anyone is manufacturing solar panels, microchips, all these, any techniques, it has to be manufactured in a completely sterile environment. So now we are concentrating majorly on the biopharmaceutical products. So when we talk about, manufacturing all these products, any medicine or medicine related things. So these processes have to be produced in a completely sterile aseptic process, aseptic room. And during this process, say for example, if we talk about manufacturing of monoclonal antibodies. Have you visited? I think in Biocon you do that, right? You have monoclonal antibodies. You produce antibodies by cell culturing. You visited the facility, right? The cell culture facility. Now,
No, ma'am.
you haven't visited the cell culture facility there.
No.
Okay, maybe I think in the future you might visit the cell culture because it's completely sterile. They will not allow anyone and everyone to visit there. Maybe one person or two people at a time, they will allow you, not more than, they will not allow you in a group there. Okay, say for example, now we have this production of monoclonal antibodies and it starts from a basic cell culture technique. Now, cell culture has to be maintained in a complete sterile aseptic environment. And we have to keep monitoring for contamination. Contamination can be caused by anything. It can be caused by bacteria and it can be caused by mycoplasmas also. Now, majorly mycoplasmas are the main, contaminating agent that can cause, these extra products, like they can produce a proteolytic enzymes. They can destroy these proteins. So these monoclonal antibodies are proteinaceous in nature. So these proteolytic enzymes, they can degrade these proteins, right? They can degrade the antibodies. And already, I think, I remember listening to someone's lecture on pyrogens. What are pyrogens?
These are the
Endotoxin.
Pyrogens are organisms that produce endotoxins. Okay, now you need to be very, very careful. We don't want any endotoxic substance in the drug that we are manufacturing. And we don't want any mycoplasmas. So mycoplasmas, it's very difficult to identify the mycoplasmas. You cannot see them under a normal light microscope. Okay, it has to be stained. And by the time you stain it, your cells will be lost. Okay, mycoplasmas, they change the growth characters also. Okay, they completely, they cause the alteration of DNA as well, of whatever cells that you are culturing. So all these, so we have to maintain complete sterility as well. And a few examples of these contaminating agents. And also how do these contaminating agents enter into these culture rooms, into aseptic rooms? Any idea?
Material, person.
Equipment.
Mainly it is through people. Okay, mainly whoever is working on the culturing of those cell lines. So the major thing that we have to, be very, very careful is we have to prevent contamination. So these microorganisms, now they have the tendency to bypass any kind of external physical barriers. Like, for example, skin, they can bypass the mucous membrane, they can bypass the digestive system. Okay. And many microorganisms, it's very difficult to differentiate between what microorganisms are, which kind of microorganisms are, harmful, or which are not harmful. Okay. And also, some people will have adverse side effects as well. So some people may be immunodeficient. So immunodeficient is people who cannot, even a simple bacterial infection, it can be deadly for them. Okay, if it is an Enterobacter or if it is Klebsiella infection, nowadays it's becoming very common because most of these bacteria, they're all becoming resistant to antibiotics. So how do we maintain it? I think previously Veeresh sir also told you about the process of sterilization. Right? So we take some precautionary measures to control the growth of these microorganisms, like dry heat, moist sterilization. Then you also have terminal sterilization. So in biopharmaceutical companies, we use this process, majorly use this process called as terminal sterilization. So what is this terminal sterilization? And do you think that all these biopharmaceutical molecules can be subjected for autoclaving? Can we do that? Definitely not, because this is what most of the pharmaceutical molecules that you manufacture, they're all heat labile molecules. That means they can get degraded on heating. And in an autoclave, what is the normal pressure and temperature that you keep for sterilization? It is very high, right? 121, what is that? Psi or 121 degrees, 15 psi, right? All forgot. Huh? Last week, what sir did?
No, ma'am, remember.
You remember. This you've been doing it from college, no? Okay. And so this is, so we need to prevent. So this terminal sterilization process is generally done when a finished product, okay, this is sterilized using heat. Now, generally, all these molecules, biomolecules, they are avoided. So we have different sterilization processes for that. I think that someone else is going to be handling those topics. Now, apart from these microorganisms, that is bacteria and fungi, mycoplasmas, we also have viruses, right? So viruses are very, very small parasites, you can call them. And they're the most simplest organisms, but they can be very, very deadly, right? They can have either DNA or RNA as their genetic material, and they only have a protein code. And these viruses, you know, they're very, very host specific. They cannot infect any Tom, Dick, and Harry. Okay, they cannot infect any cell lines. They only prefer a particular host for them. Now, these viruses also can be a contaminating agent. And all these viruses also have to be identified and completely sterilized. They have to be killed to prevent any other contaminations during our process. And apart from viruses, we also have one more very, very important microorganisms. These are prions. Prions, they are again infectious agents. They are made up of proteins only. They do not have any kind of associated nucleic acids. And these prions, they are referred to as prions because they are misfolded proteins. And you know that prions cause a particular kind of disease called as scrappy disease in sheep and Creutzfeldt Jacob disease in humans. Now, these prions can also be highly contaminating agents during our process of cell culturing. So if we are deriving any cells from sheep, because generally when you talk about cloning and other things, prions are the infectious agents which cause major contamination. Okay, because the animals are the sources of cell lines, blood. So you're going to be using all these sources for culturing. And slowly, all these cultures are the ones that you're going to experiment with, and you're going to use them for the bioprocessing for any other manufacturing process. So now, how do we, so when you start with the manufacturing of any drugs substance, so when you talk about drug manufacturing process, you have studied certain standard operating procedures and all that. Now, all these SOPs that you follow in the industry level, now these microbiological processes, they start at the research level. Majorly, they're all into research level. So when you start with the preparation of any process that starts with the manufacturing of a drug, now you need to maintain or achieve microbiological control. So in various phases, you need to check the containers that you are using for the manufacturing of drugs. You need to check the sterility of those containers. And when you're preparing a single or a small number of containers or any substance, now you have to allow this, you have to have a very low bioburden. What do you mean by this word bioburden?
Total amount content of microorganism present.
Yes, yes. So bioburden is nothing but the total microbial count has to be very, very less. Okay. Can you just hold on for a minute? Yeah, sorry about that. Okay, so this bioburden, so we need to have this process which is happening. Okay, so we need to analyze, we need to document what are the different microbes that are present. So for this controlling of this bioburden, this is called as bioburden control strategy. So we need to maintain this because typically in a lab, you will have XYZ number of or XYZ type of microorganisms. Okay, now we need to keep a track of these microorganisms. We need to make sure that they are not toxic in nature or they do not produce any toxins, and it should be easy to eliminate them. And if they're not easy to eliminate them, we have to come up with other things to reduce the number. Now, in reality, I told you it is not possible to have a completely sterile environment for the production process. So now, so all the drug products, they all have to be sterilized and they have to be placed in a, when they're placed in a final container, they have to be completely sterile. Why do you think that all these drugs have to be manufactured in a sterile condition? These drugs are used for what?
Since it is a parental drug, so it should be produced in a sterile condition.
It is
These
given
drugs
to
are going to be supplied to
patients.
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 Patients are already having a weak immune system. Okay, these drugs are just given to enhance their immune system. Now, already if it is contaminated, then there is a very high risk of this patient, kicking the bucket and, okay, directly taking one ticket to wherever. Okay, so this is the reason why we need to manufacture drugs and always in a sterile environment. And the next term that I was asking you is aseptic. So so we always have to manufacture these drugs in an aseptic environment. That means that these aseptic processes, they're used for those drug process that has to be sterile, but cannot be subjected for terminal sterilization because these drug products they're all heat liable and they will lose their effect when they are sterilized. Okay, so that is why these drugs have to be manufactured in a complete aseptic environment. And anexic, this is another term. So anexic culture. What do you mean by this word anexic? Sorry, not anexic, it is axenic. So axenic is they are free from any kind of microorganisms. So all biopharmaceuticals, they are all intended to be axenic cultures. So only supposing you are deriving any drug product from cell lines like monoclonal antibodies or polyclonal antibodies, so they only should contain the cell line desired characteristics, and they should not have any microorganisms. So these are the three major types of your contaminants. Okay. So what are the types of contamination that you can usually find? So contamination means it's a presence of unwanted substances that's going to affect the stability, the strength, and purity of the drug product. Now, these contaminants, they can be transferred into any kind of sterile components, and basically all these are grouped into two categories, major categories: particulate or microbial contaminations. Now, both these types, they are not visible to the naked eyes. You cannot see them directly. Particulate contaminations, you cannot see. Now, when do you think you can see these particulates? Particulate can be dust. Dust can also cause contamination. Correct. So when can you see dust? What is that effect called as? I mean, when you were young, you would have seen this. When sunlight passes.
Tyndall effect, ma'am.
Tyndall effect. So in Tyndall effect, you can see the dust, right? You can see all the dust particles flying in the air. Now, so particulates are basically small bits of any matter. It can be, these are microscopic in dimension, and you can categorize into different sizes. So generally, these particle, which is smaller than the human eye, these are, say, approximately around 25 microns in size. And so this diagram that you can see here, this is the dark green one is actually the section. That's the measurement of a human hair. And the small dot that you see inside the light green circle, that is 1 micron size particles. So they're very, very tiny. So these tiny particles also can cause contamination. All right. And these particulates, they can be made up of either organic or inorganic materials. And they're usually found in all types, that is gases, liquids, and solids. They can be suspended or they can be settled material. So whatever material you're using for the drug manufacturing process also has to be in sterile condition. They should be free from any kind of particles. So in gas, in the gas that you're using, see, gas is generally used for to maintain your culture. So if you're using cell line, we need to have one carbon dioxide in the incubators. When the cell lines are cultured, you culture them in huge incubators, right? Or you use fermenters. Most of the cases, you will use fermenters. So the gas that you are supplying inside these fermenters, they have to be sterile in nature. They should not contain any kind of particles. So, so any particle which is suspended in gas, it is called as an aerosol, or it causes airborne contamination. In liquid, it is suspension, usually in the media, culture media that you're using for the cell line culturing and all that, they'll be settled at the bottom. It can be, okay, and in the solid, it can be any kind of matter. So all these are very harmful for the final product. And these particulates, so if you're using paper, so in the lab, I'm not talking about in industry level. So generally in the lab, our PG, UG or any kind of research lab, we always tend to wrap these glasswares in newspapers, and we will sterilize them. We will keep it for autoclaving. So whatever culture vessels that we are having, we generally wrap them with paper, newspaper. Okay, now these particles, sometimes they can be harmful. So the cellulose fiber, they can interfere with your drug product. And sometimes when you are sterilizing glasswares, they can break. When you are, see, if it's a very low quality glassware, they can easily break. So those breakages, those glass particles also can cause contamination. Then aluminum particles. So you're using aluminum caps, right? For when you are manufacturing some liquid drugs in the syringe and all that, in vials and all that. When you're using aluminum, caps, those aluminum also can cause some kind of metal contamination. And the gowning that you are wearing, so they will have some kind of fibers. So those fibers also can cause contamination. And the main product, the main contamination is human hair, skin. So in a clean room, these are the major contaminants: hair, dead skin. And you are aware that you are not supposed to talk also. In a complete sterile environment, you should not talk, because talking also is going to release some microorganisms. Your mouth is the most in your body. It's not the skin, it's not the skin surface, or it's not your face, not your hair. That is a highly contaminant region. Okay, the most highly contaminated part of your body is your mouth. Okay, so careful. So your nasal area is the major source of these microbial contaminations. Now, supposing if there is any kind of microbial contamination, how fast can these microorganisms multiply? Bacteria are the ones that replicate once every 20 minutes. And those who wanted to go ahead with their further studies after, working for us, we'll have a few students who want to work for a few years in industry. And then if they want to go into research, or if they want to continue into go for higher studies, they have to write an entrance exam. Now, one favorite question in most of the entrance exams is, I'm having X number of bacteria. After 20 minutes, this X number of bacteria multiplies. So what is the number of bacteria? So they will give you some options. You need to tick the correct option. So you have one bacteria. One bacteria takes 20 minutes to replicate. Next 20 minutes, how many bacteria would I have? So bacteria multiply exponentially. That means one bacteria at the end of 20 minutes, I will have 2 bacteria. 2 bacteria will multiply into 4 bacteria. 4 into 8, 8 into 16, 16 into 32. So it is always going to be exponential. Now, generally, I'm not going to have 1 bacteria. That's not possible at all. I will have number of bacteria. Now, if I'm considering say, for example, if there is some kind of contamination, 9 a.m. I'm entering into my lab. I'm carrying so many number of bacteria. It is getting, by mistake, I have contaminated that area. Now, by the end, 5 p.m., I'm closing my lab and going. So we will not know. So I will have around 16 crores of bacteria by the end of the day. And most of the bacteria, so in general, clean rooms or a most of these bacteria, they're all gram-negative in nature. That means they are all very pathogenic in nature. Most of the gram-negative bacteria are all pathogenic. And These are some commonly found microorganisms in a clean room. So you will have Staphylococcus species, Micrococcus species, Bacillus species, and the sources. So Bacillus you can easily get from soil. So any dust particles, if you are walking, okay, you can have Bacillus on your shoes. And all these are some sources of contamination. So your human hair, okay, all your clothes, when you're sneezing, when you're coughing, and also one thing, the mask that you wear, definitely these microorganisms cannot be blocked even if you're wearing a mask. And so in your nose, in your spit, scalp, forehead, armpit, hands, you have these many number of microorganisms. So we need to be very, very clean. So how do we control the contaminants in a clean room? One thing is you can control by using air supply. So air supply using HEPA filters. I think you already studied that, right? In a clean room, you have continuous supply of air, and whatever air that is supplied into that clean room area, it is in a sterile form. So we are using HEPA filters, which filter out all the particulates. It can be organic, inorganic particulates. So no spores, minimal spores will be there. So spores are nothing but these are the reproductive nature of microorganisms. So it can be spores from fungi, or you can have spores from bacteria also. So all these will be removed using HEPA filters. And any equipment that you're using, you can always sterilize them using dry heat, using gamma radiation. You can completely radiate the room with gamma rays, or you can spray with ethylene dioxide, or you can use hydrogen peroxide. So hydrogen peroxide is majorly used in most of the places, and filtration is also done. So these are some common methods of sterilizing. And how do we validate that whatever sterilization process that we have carried out, is it properly done or not? How do we check? Because that is another main thing, right? So once the sterilization process is done, we need to check whether it is completely sterile or not. And endospores, or some endospores, you cannot kill them. However, or whatever methods you use for sterilization, you cannot kill them. So we need to validate the process. So how do we do that? So in an autoclave, we are going to run a few cycles. We are going to check for certain parameters. So we are going to check for this bioburden reduction. So if it is achieved or not in an autoclave, we are going to keep a culture and keep autoclaving it until it is dead. Then we can use some biological indicators for this reason. So we are going to have a known amount of microorganisms, and these known amounts of microorganisms, we are going to expose them in a completely sterile condition. And that means what? In a complete sterile environment means what? So we are going to check whether these microorganisms, whether they're growing or they're getting killed. So if there is an increase in the number, that means that if the sterilization process is not done properly. Okay. Now, again, we're going to keep monitoring the temperature, the pressure, the autoclaving cycles that we are using, and also the load that you add. See, in a normal, in your house, okay, if you're using a washing machine, there is a particular amount of load that you have to add. So this many kgs of clothes only I can add, and I can wash them. Similarly, even in an autoclave, I have only these many, items or these many number of glassware that I need to autoclave. I cannot completely load the autoclave and I cannot sterilize it. If there is some place there, and sterilization, it takes some time. You don't have time. At least you need a minimum of 1 hour. I don't have that 1 hour. So what you try to do? You try to add excess loads into your instruments for sterilization. And what this does, what happens is there's improper sterilization that is achieved. So you will not achieve your proper sterile state. Okay. So once this is validated, so once after you have validated the clean room. Now, when I'm talking about clean room, now there are different standards of clean room. Do you know that? You have clean room 1, you have clean room 100, you have clean room 10,000, etc. So they're all related to this ISO. They have given some ISO numbers. Now these clean room, where the numbers, the clean room 100 or 10,000, that means that for 1 cubic meter, I will have only 100 particles. It's not possible. See, I told you it is impossible to maintain a completely sterile environment. So I can allow some amount of particles to be present. Okay. So during the manufacturing process, in clean room 100, I should have only 100 particles. It can be any kind of particles, only 100 particles per cubic meter. If I have more than 100 particles, that means that room is not sterile. I need to clean the room. I need to sterilize the room. Now, how do I prevent this process? How do I prevent? See, I cannot maintain. I have to check whether that room is completely sterile in nature or not. So I need to be taking some precautions. And we know that people are the most major sources of contaminants. And we keep shedding dry skin, dead skin. And if you have oil in your hair, the oil can also accommodate a lot of microorganisms, right? And every time you move or every time you talk, you're going to carry so many microorganisms and you're going to shed so many microorganisms. And at most, a person can shed approximately 10 to the power 9 cells per day. And so to prevent all the shedding of microorganisms, we wear clean, sterilized clothes. That is your aseptic gowns. So you're going to wear masks, gloves, goggles, and boots. I think we see Dodda Basaya always in sterile, in aseptic gowns, right? He's not there, Dodda Basaya, in that session. And these gowns, they will not provide 100% protection. These microorganisms, they can penetrate these fabric and they can come out. They can come out from our own body. Obviously, they can come out of aseptic gowns also. Okay, so yeah, so this is what I was talking to you about. So we have different classes where how these environments we have to maintain them. So a clean area, we have 10 class 100, class 10,000, class 100,000, and we have controlled unclassified. And these are all the ISO designation. So 5, 7, 8 and all that. And for class 100, we need to have a fully aseptic gowning. We need to wear sterile gowns, hoods, okay? Then double gloves, masks, and all that. So class 10,000 also, it is the same thing. And class 100,000 is, we are supposed to wear non-shedding. We have to wear 2 suits, 2 gowns. We need to, make sure that they are tight at the wrist, at the ankles, at the shoe covers and all that. I think COVID or you would have seen everyone wearing the PPE kit. Most of them, all these healthcare workers, to prevent the entry of all these organisms. Okay. So any person who is unwell should not enter the clean room. Anyone having any kind of infection, gastrointestinal infection, or any kind of lesions on the skin, or if they've undergone some, some treatment. So all these people should not come to work for a few days. Okay. And anyone, if you've undergone, I think girls, most of them, they undergo these peeling and all that, no, skin peeling, all those. So all these people should not work on a, should not work on in a clean room. And there are some practices that we need to follow for aseptic gowning. We need to wash our hands with soap and water before entering the gowning area. You have to always keep your nails trimmed. Do not wear nail polish. Okay, your nails has to be very, very clean, and you're not supposed to wear makeup also. Cover your face with masks. Make sure all your hairs are not loose, they're all tied up, and always wear gloves. Keep sterilizing your gloves, that is your hands, wearing your gloves with isopropyl alcohol, 70% isopropyl alcohol. And always you need to make sure that the gown fits you properly. You cannot wear a very loosely fitting gown. Okay, so these improper size gowns also can cause some problems. It can also lead to contamination. And talk minimum. You can use sign languages, because you know that when you're talking also, microorganisms can spread. The more you talk, the more number of microorganisms I'm spreading. So avoid the coughing, sneezing, and all these. So these are some rules that you can follow. And also, we need to control physical movements also. So whenever you have an open cell culture, so if you have a cell culture system that is left in the open, make sure that movements are reduced. Okay, because the more you move, the more you're causing a little stir in the environment. So these stirring can cause imbalance in the particulates also. And the environment, you can keep the environment clean using HEPA filtered HEPA filtered air. Then all the equipments that you're using, make sure they are clean. And yeah, in the you have different levels of biosafety cabinets. So when you're working with a cell culture, you have level 1 biosafety cabinet, you have a level 2, you have a level 3 biosafety cabinets. Each safety cabinets are designated for specific tasks. So in the biosafety cabinets, make sure that it is cleaned periodically. So by the end of the day, so today I'm working on so-and-so culture, cell line. I need to make sure that by the end of the day, the safety cabinet is clean, there is no contamination, all that. And always plan your work. Now, planning is done to, to minimize any kind of physical movement. So once the operation starts, say it can be any kind of transferring of events, or if you're transferring the vials from one room to another room, you need to make sure that you are not moving there. It is completely moving in a sterile environment. So any kind, all these, whether it is a biosafety cabinet, you make sure that you're using a vacuum cabinet, and so that it doesn't pull any kind of contamination from outside, and you can perform any kind of aseptic manipulations and above the waste level. So a biosafety cabinet, it covers from your head to your waist. You need to sit, you cannot stand and work. I think in this previous picture, you would have seen, this is a biosafety cabinet. Can you see that there's one person standing, one person sitting there, another person standing? It is not supposed to happen like this. Okay, so you are supposed to sit, and only your hands should be inside the biosafety cabinets, not your entire self. Now, sometimes when you are just a fresher, when you're learning how to work in a biosafety cabinets, we won't be confident enough. So we are going to push ourselves into the biosafety cabinets, and you're going to carry out all your work there. So that has to be avoided. Okay, and all, if you're using any belt, pads, all these should be sterilized. And yeah, so plan everything, make sure that you are not moving anywhere near the biosafety cabinet. You just have to sit in one place, and you have to carry out all the work there. And you have to clean any equipment, any material that you are bringing out of the biosafety cabinet, you need to clean it. And when any equipment you're taking inside the biosafety cabinet also has to be sterilized. So inside, outside, you always have to sterilize them. And you can use any kind of material that you can keep cleaning them, and without compromising that material. You can use metals. Okay, if you're into culturing, all your forceps or your inoculation loops, they're all made of metals. Okay, so you can sterilize them a number of times, nothing is going to happen to them. And in the biosafety cabinets, you are going to separate clean waste and dirty waste. So, say for example, when you are wrapping them, wrapping any glasswares, you need to remove the wrapper, whether it is plastic or paper, it doesn't matter. So all these are going to be dirty waste. And not only that, if there is any contamination, that also will be dirty waste. You are cleaning the biosafety cabinet. So when you start your work, you always have to make sure that the biosafety cabinet is clean. You're going to clean it with alcohol or the hydrogen peroxide or whatever, anything you can use for cleaning. Once you clean the biosafety area, the waste, you're using some paper or cotton, sterile cotton for cleaning. That is again waste. So you need to know where you're going to place the waste materials in the biosafety cabinet. I cannot place it near the biosafety cabinets. They're lined with HEPA filters. Okay, at the back or at the top will be lined with HEPA filters. So you have to make sure that I don't interfere the air circulation. So I need to keep these waste somewhere. And there's another problem now. Once I bring my hand outside the biosafety cabinet, I have to, again, when I put my hand back into it, even though I'm wearing gloves, I'm in a clean room, people might think, though I'm in a clean room, once my hand is out, it's okay, I don't have to sterilize my hand. That is totally wrong. You need to again sterilize your hand, and then only you are supposed to put your hand inside the biosafety cabinets. So again, it's all up to you. So the more aware you are of the cleanliness in a biosafety cabinet, you can avoid the contamination. And So again, yeah, limit talking, work in an unhurried manner. So when you are sitting in a biosafety, front of the biosafety cabinet, make sure that all the things are ready near your hand. Okay, I don't have to keep removing my hand every now and then. So make sure everything is at the reach of your hand. Okay, then my friend is there, my colleague is there. I should not keep asking my colleague to go get one forceps or to go get one needle. Okay, because I have to minimize talking there. And so make sure that it is well planned, less movement, and you have to refrain from getting any all these papers, cardboard and all that. You need to avoid using them. And make sure that you are completely gowned. The room is airlocked, because once it is airlocked, there is no, inlet or outlet of air there. Air is just getting circulated. So both the airlock, so if make sure that you don't open two airlock doors simultaneously, because there'll be a rush of vacuum there. Okay, so you can control. Yeah, I know it's time up.
Okay.
Yeah, I'll stop in another 5 minutes and I'll leave. So how do we control this infection, or how do we control all these contamination? So we can control them by using some kind of disinfection. Okay, and we know that 1 micron of anthrax spore can take approximately 20 minutes to float and settle on, at a distance of 1 meter. So it just takes 20 minutes for 1 spore of anthrax. And anthrax is a very, very, very harmful bacteria. Okay, it's a deadly disease. It causes a deadly disease. So be careful. And what are the cleaning agents? What do you mean by cleaning and disinfection?
Ma'am, cleaning like sanitization,
Disinfection?
Huh?
Disinfection?
Completely removing the microorganism.
Okay. So cleaning is it's the physical removal of any particles from the surface. You can either mop, wipe, or brush. Disinfection is complete elimination of harmful microorganisms. So disinfection does not achieve 100% sterilization. Okay, you need to use a whole load of disinfection techniques for achieving 100% sterile environment. So all that, I think someone else will be doing it. So these are some examples of disinfecting agents I have given here: alcohols, aldehydes, halogens, phenolics, and other agents. Okay, and where can you achieve all these disinfection? So in a class 100 area, phenols can be used. So every month you can subject that area for for any phenolic activity, or you can you can phosphorucide. That means it's going to kill all the spores. So walls, you can spray it with some phenol material, and all the curtains, the surfaces, you can wipe them. Okay, and class 10,000, same, you can use the same process. And for wiping, you can use isopropyl alcohol. And how often should you clean? So everyday cleaning can reduce the contamination. Okay, so again, I have given you. And aseptic clean rooms. So we need to keep monitoring. So monitoring. So this I will start in the next class. So in an aseptic clean room, how do I monitor all these contaminating agents? So what are the different techniques of environmental monitoring, and how can I prevent this sort of contamination. So my slide continues with the next class. It's the same slide. I think you already have the slides, right?
Yes, ma'am.
Yeah, okay. All right. So I will see you all on Tuesday morning.
Okay, ma'am.
Okay.
Thank you.
I hope you all didn't sleep for my class. The microorganisms. All are tired. So, okay. So have a nice lunch. And does anyone have any questions related to whatever I just spoke to you about?
No,
No, that means you've understood everything.
it's already done, ma'am. Which you thought today, it's already done for us.
Lakshmi ma'am already did.
Not Lakshmi, ma'am. In functional visit.
Okay. All right. So I will see you on Tuesday then.
Thank you, ma'am.
Thank you,
Okay,
ma'am.
Thank you, ma'am.
bye-bye.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
ma'am.